Hello everyone, welcome back. Today we will discuss one important topic in molecular biology, that is transcription. In this lecture we will discuss about introduction to transcription, then we will discuss types of RNAs and the mechanism of transcription in prokaryotes. So let's start. Firstly, what is transcription? It is the process by which DNA conveys its message to form mRNA or other types of RNAs is known as transcription. So process of formation of transcript, that is RNA, is known as transcription. It is catalyzed and scrutinized by enzyme which is known as RNA Polymerase. जो ये सारा काम होता है, it is done by this enzyme, that is RNA Polymerase. So this polymerase enzyme, that is RNA Polymerase, helps in formation of this RNA, that is mRNA, from one strand of DNA. It occurs unidirectionally, in which RNA chain or transcript is synthesized from 5' to 3' direction. So ये जो synthesis होती है RNA की, that is in 5' to 3' direction. This is a very important concept. And moves out from nucleus and carries out translation in cytoplasm. So DNA conveys its message through this messenger RNA to the particular protein, which are synthesized in cytoplasm. Genetic material in some viruses. This RNA acts as genetic material in some viruses, like we have Rheovirus. Rheoviruses contain double-stranded RNA, then retroviruses like HIV. So these all viruses contain RNA as their genetic material. Basically, this is the central dogma of biology. It means it is a unidirectional flow of information from DNA to proteins through this mRNA, messenger RNA, which is synthesized inside the nucleus in case of eukaryotes, which is carried out to the cytoplasm to synthesize a particular proteins. So this whole process is known as central dogma of biology. Next, what is RNA? Generally, RNA is a single-stranded structure which contains ribose sugar, which also act as genetic material in some viruses like Rheoviruses. Base composition of RNA does not follow Chargaff rule. This is a very important difference between DNA and RNA. So जो base composition है RNA की, वो Chargaff rule को follow नहीं करती है. That is, जो ratio है adenine का or means purines or pyrimidines का, that is not equal. So there are 4 common bases which are present in RNA, which are adenine, guanine, uracil, and cytosine. So thymine is replaced by uracil in case of RNA. Here thymine is absent. Then sugar in the RNA is ribose type. So these are some differences between DNA and RNA. This picture shows the comparison between DNA and RNA. In case of DNA, it is a double-stranded structure, and इसमें जो sugar present होती है, that is deoxyribose sugar. Why it is called deoxyribose? Because in the second carbon atom here, oxygen is absent. So that's why this is known as deoxyribose sugar. In addition to this, it contains a base which is thymine, which is present in case of DNA, while in case of RNA, it is usually single-stranded structure. और इसमें जो sugar present होता है, that is ribose sugar. Here we can see second carbon atom पे present होता है oxygen. As compared to DNA, RNA is less stable because it is single-stranded, और इसमें oxygen present होता है, that helps in oxidation. So that's why it is not that much frequent genetic material in all the organisms, except with a few exceptions in case of viruses. So these are some different. Next comes transcription unit. It is the functional segment of DNA that undergoes the process of transcription, is known as transcription unit. So transcription is highly selective process. ऐसा नहीं है कि सारा का सारा जो DNA है that will undergo transcription. A particular portion of DNA will undergo the process of transcription. So it is highly selective process. And a specific segment of DNA is transcribed, which is known as transcription unit. DNA का वो segment जिसमें transcription होगी, उसे transcription unit कहा जाता है. In eukaryotes, a transcription unit typically carries the information of just one gene, and it is termed as monocistronic. So this is an important point. In case of eukaryotes, only a single gene carries the information for a particular protein. So that transcription unit is known as monocistronic transcription unit. So in this picture, we can see here, this is eukaryotic mRNA, which contains a gene for a single protein. So this type of transcription unit is known as monocistronic transcription unit. In case of prokaryotes, a set of adjacent genes is often transcribed as unit, termed as polycistronic transcription unit. और prokaryotes के केस में जो mRNA होता है that contains information for more than one protein. So this type of transcription unit is known as polycistronic transcription unit. The immediate product of transcription in case of eukaryotes is primary transcript, और उसे hnRNA भी कहा जाता है, that will undergo the process of processing, RNA processing, that we'll discuss in case of eukaryotic transcription. Next, only one strand of the transcription unit is transcribed. जो DNA का double stranded structure होता है उसमें से एक ही strand transcription show करता है, means that will undergo transcription process and form the RNA, single stranded RNA. Therefore, the transcript is identical in sequence with one strand of DNA, which is called as the coding strand, and the complementary to the other strand is called template strand. So out of these two DNA strands, one strand will act as the coding strand or one strand will act as a template strand. So वो strand DNA का जिसके sequences, base sequences, complementary sequences का RNA बनता है, so that strand is known as template strand or anti-sense strand, while जो RNA है उसी की तरह same base sequences present होते हैं DNA के दूसरे strand में, जिसे कहा जाता है sense strand और coding strand भी. In addition to this, it is also known as positive strand. So the coding strand is also known as sense strand, or it is represented by a plus symbol strand, while the template strand is known as anti-sense strand, or it is represented with the minus symbol. Any region of the DNA double helix could be copied into two different RNA molecules, one from each strand of the two DNA strands. So जो DNA के दोनों strands है वो दो RNAs बना सकते हैं, but in reality only one strand will undergo the process of transcription. In reality only one DNA strand is used as template in each region. Next, types of RNA. Basically RNA दो तरह के होते हैं. One is genetic RNA, second is non-genetic RNA. Genetic RNA means here this RNA acts as the genetic material. It occurs as genetic material in some viruses like poliomyelitis, Ross sarcoma, which causes a cancer of soft tissue, and influenza, commonly known as flu, with single stranded RNA. So ये जो viruses हैं इनमें RNA as a genetic material present होता है. There is one exception, that is Rheovirus. Here the RNA is double stranded. यहाँ पे RNA double stranded होता है. Then we have some bacteriophages which contains RNA as genetic material like MS2, F2, R17. These are some bacteriophages, means viruses which infect bacteria. They contain genetic material as single stranded RNA. Then we in this lecture we'll discuss only the non-genetic RNAs, which are of three types. One is mRNA, then rRNA and tRNA. mRNA means messenger RNA, then ribosomal RNA and transfer RNA. So these RNAs we will discuss one by one in detail. So non-genetic RNA. There are basically three types of non-genetic RNA that we have already discussed. That is ribosomal RNA, messenger RNA, and tRNA, that is transfer RNA. So this messenger RNA helps to carry its code from DNA to ribosomes for synthesis of particular protein or polypeptide. This ribosomal RNA, it assembles amino acids brought by transfer RNA in a specific order from mRNA to make proteins made up of RNA by nucleus. So this ribosomal RNA is synthesized inside the nucleus, जिसे कहा जाता है NOR, that is nuclear organizer region. Then next comes transfer RNA. It helps in transport of specific amino acid to ribosomes for a particular protein synthesis and carries anticodons, which are complementary to the codons of mRNA. So by reading the codes of mRNA, this tRNA helps in accepting the particular amino acid and helps in formation of polypeptide. So these are the three basic types of non-genetic RNAs. having different structural features and specific functions. So these three RNAs have different structural features and specific functions. Firstly, we'll discuss about ribosomal RNA. It is found inside ribosome and called ribosomal RNA, differ in size and base content from other types of RNAs. इसमें जो size है और base content है वो vary करता है among other RNAs. And it is rich in guanine and cytosine and constitute about 70 to 80% of the total RNA content. Just maximum RNA present होता है किसी भी cell में, there is the ribosomal RNA that is 70 to 80% inside the cell. And it is a single polynucleotide strand, extremely coiled and folded with the ribosomes. In prokaryotes, based on sedimentation coefficient, ribosome is called 70S and consists of 50S subunit and 30S subunit. So 50S subunit is the larger subunit and 30S subunit is the smaller subunit. It further consists of different types of rRNAs, like we have 5S RNA and 23RS NA, which forms the largest subunit of prokaryotic ribosome, and then we have smaller subunit which contains 16S RNA. Here S stands for sedimentation coefficient. In case of eukaryotes, there are again two subunits which are 60S subunit and 40S subunit, which combine to form 80S subunit. Here 40S subunit consists of 18S rRNA and 18S, and 16S subunit consists of three types of rRNA, which are 28S RNA, 5.8S RNA, and 5S RNA. So these are the subunits of this larger subunit of ribosome in case of eukaryotes. Next comes subunits of prokaryotic and eukaryotic ribosomes. As we know that in case of prokaryotic ribosome, it is of 70S ribosome, which is present in case of prokaryotes. So it consists of two subunits which are made up of 30S and 50S subunit respectively. So 30S subunit of prokaryotic ribosome contains 16S RNA. 16S RNA is further made up of 1542 base pairs plus 21 proteins. This is the chemical composition of this 30S subunit of prokaryotic ribosome. That is a smaller subunit. Then the larger subunit, that is the 50S subunit of ribosome of prokaryotes, contains 23S rRNA, which is made up of 2904 base pairs, and 5S rRNA, which is made up of 120 base pairs along with 31 proteins. So this is the chemical composition of larger subunit of prokaryotic ribosome. In case of eukaryotes, ribosomes are heavier with sedimentation coefficient of 80S, while in case of prokaryote it was 70S. The two subunits consist of 40S and 16S rRNAs. The 40S, that is smaller subunit, contains 18S rRNA, which is made up of 1874 base pairs plus 33 proteins. This is the chemical composition of the smaller subunit of eukaryotic ribosome. Next comes larger subunit, that is 16S subunit, which is made up of 3 rRNA molecules like 28S rRNA, then 5.8 rRNA, and 5S rRNA, which are made up of 4718 base pairs, then 160 base pair and 120 base pair, and it contains 49 proteins. So this is the chemical composition of larger subunit of ribosome of eukaryote. And next comes messenger RNA. So DNA transfer its information with the help of this rRNA, which is known as mRNA, to form a particular protein in the cytoplasm in case of eukaryotes. And this RNA, which carries this information from DNA to a particular protein, is known as mRNA. So it is basically a single-stranded structure and bears exact copy of nucleosides. In DNA, one strand referred as template strand. Template strand is the strand which contains from which the RNA is synthesized. So that parental strand जिनके कोड्स को देख के RNA synthesize होता है, that strand is known as template strand, और इसे antisense strand भी कहा जाता है, is transcribed to form this mRNA and makes about 3 to 5% of total RNA. जो total RNA present होता है सेल में, उसका 3 to 5% mRNA होता है. And nucleotide sequence in mRNA are in triplets form, जिन्हें कहा जाता है codons. and size of this mRNA varies. Length of E. coli mRNA ranges from 900 to 1500 base pairs. So this is the approximation length of E. coli's mRNA. In case of prokaryotes, mRNA are short-lived and generally polycistronic, that we have already discussed. It means it codes for more than one protein. In eukaryotes, mRNA is generally transcribed as large precursor molecule, which जिसे कहा जाता है प्राइमरी ट्रांसक्रिप्ट या फिर heterogeneous nuclear RNA, that is HNRNA. ये जो RNA है, it have to undergo the processing process, जिसमें इसकी modification होती है. Modification include 3 steps. So this eukaryotic HNRNA और primary transcript undergoes post-transcriptional modifications, जिसमें आता है splicing, polyadenylation and capping. So these 3 processes occurs during the process of this post-transcriptional modification in nucleus and then transported to the cytoplasm where it is translated, means it carries information part of a particular protein. Because this primary transcript, which is synthesized from DNA, it contains both coding regions and non-coding regions. Coding regions are known as exons and non-coding regions are known as introns. So before it becomes functional, we have to remove, the cell has to remove this non-coding sequences. Introns को पहले remove करना पड़ेगा, उसके लिए इसमें होती है splicing. It forms a structure which is known as spliceosome, जिसमें help करते हैं snRNAs. So these helps in this removal of non-coding sequences, जिन्हें introns कहा जाता है. And after splicing, we will get the functional segment. Before this, there is addition of 7-methyl guanosine group at the 5' end, जिसे capping कहा जाता है. And after splicing, there is addition of the polyadenylation. That is, बहुत सारी जो adenine groups हैं, लगभग 200 adenine groups, they are added at 3' end. And this process is known as tailing. इस process को कहा जाता है tailing. Average eukaryotic mRNA is about 1000 to 2000 base pair long. The 5' terminus is capped by 7-methyl guanosine triphosphate. So this is known as capping, और इसका जो function है, it helps in recognition of mRNA by translating machinery, that is ribosomes. So जो ribosome है वो इस mRNA को identify करता है due to presence of this cap, which is made up of 7-methyl guanosine triphosphate. Cap is followed by initiation codon, that is AUG. It codes for amino acid which is known as methionine. Other end possesses termination codons, जहाँ पे transcription means protein बनने बंद होंगे, वहाँ पे special codons present होते हैं जिन्हें कहा जाता है UAA, UAG and UGA. 3' end of mRNA usually has attached polyadenylic acid sequences, which ranges from 200 to 250 in number. So this process is known as tailing. It helps in stability, to maintain the stability of this mRNA. Next comes the third type of RNA, that is transfer RNA, abbreviated as tRNA. It is also known as adapter molecule or soluble RNA and transfer RNA. Cytoplasmic RNA, this is a cytoplasmic RNA, means it is found in cytoplasm and not bound with ribosome and is soluble in 1 molar NaCl solution. So that's why it is also known as soluble RNA and is the smallest RNA among all the types of RNAs. It forms about 15% of total RNA. And it is smallest among all the polynucleotides, that is 73 to 93 bases. In eukaryotes, tRNA is synthesized by RNA Polymerase 3, while mRNA is formed by RNA Polymerase 2 in case of eukaryotes. 3 ribonucleotides, that is CCA, are added to its 3' end after separation from DNA template. So its 3' end contains these specific bases, that is CCA, which is the unique feature of this tRNA at 3' end. At 5' end always ends with either purine like G or with cytosine, that is pyrimidine. So 5' end पे या तो guanine bases present होंगे या फिर cytosine bases present होंगे. It contains certain unusual bases like pseudo-uridylic acid, ribothiamidilic acid, or inosine 5-methyl cytosine. So these are some unusual bases which are present in this tRNA. Primary nucleotide sequences allow extensive folding and helps to generate a secondary structure that appears like clover leaf. So जो secondary structure होता है tRNA का, that is clover leaf-like structure, and this structure was presented by Robert Holley, or explained by Robert Holley in 1964. And its tertiary structure is L-shaped. That is a three-dimensional structure, L-shaped structure of tRNA. So this picture shows the secondary structure of tRNA, which is the clover leaf-like structure, and this structure, which is the three-dimensional structure, which is L-shaped structure. So this two-dimensional structure of this tRNA. Next comes structure of tRNA. All tRNA molecules contain 4 main arms. In addition to this, it contains 1/5 arm that is known as variable arm. This is the smallest. This arm is of variable size. It is not fixed. First one is the acceptor arm. Then comes the T-C arm. Third one is the anticodon arm, and fourth one is the D arm. Beside this, they have an extra arm which have variable features. It is also known as variable. So firstly, the acceptor arm. It consists of base paired stem having 3' end and 5' end. The nucleotides at 3' OH group ends are always in the sequence of CCAOH that we have already discussed, and the 5' ends have GC rich bases. यहाँ पे GC rich bases होते हैं. So this is a structure. In 3' end, it contains these bases which are CCA along with this hydroxyl group, while at 5' terminus, it contains guanine or cytosine groups. And this structure is known as stem, and this rounded structure is known as loop. Due to complementarity, ये जो structure है ये stem की तरह structure बनता है because it contains complementary bases like G and C, A or U. So that's why it gives rise to these stem-like structures. The 3' end gets amino acylated, whereas the function of 5' end is not understood. So जो 3' end है इस tRNA का ये help करता है amino acids को carry करने के लिए from the amino acid pool in the cytoplasm. So that's why it is also known as acceptor arm. Next comes the T-C arm. It is a base paired stem and unpaired loop. Here the T-C loop. The loop bears thymine, and this psi represents the pseudouridine, that is the unusual base, and cytosine. This is a special arm, T-C arm. It is involved in the recognition of tRNA molecule by ribosome. So इसका जो काम है ये arm help करती है recognition by ribosomes. So this arm, this T-C arm, here the psi represents the unusual base, that is pseudouridine. It helps in recognition by ribosomes in the cytoplasm for protein synthesis. Next comes the anticodon arm. As the name indicates, this arm has base paired stem and unpaired loop, just similar to that of the T-C arm, that is contain anticodon loop. This is known as anticodon loop. It contains the anticodon triplets in the center of the loop that recognizes codon of the template mRNA. So ये जो anticodon loop है इसमें present होते हैं anticodons which are in the form of triplets, और ये जो anticodons है they reads the codons of mRNA which these are complementary to the codons of mRNA. जो tRNA की anticodon arm है इस पे जो bases हैं they are complementary to codons of mRNA and they carry information for a particular amino acid. It has a sequence of 3 nucleotide complementary to codons. So this anticodon carries 3 nucleotide complementary. It's 3 nucleotide sequences which are complementary to the codons of mRNA. Sometimes there is flexibility in recognition of the anticodon bases by those of the codons which differ only by the last base. Sometimes जो last base है वहाँ पे ये जो tRNA के anticodons they show some flexibility in response to mRNA's codon and they code for a particular amino acid, and this last base doesn't affect the constitution of that amino acid. So this flexibility shown by these anticodons is known as wobble hypothesis. It is called wobble hypothesis, and this concept was given by Francis Crick. Next comes the D arm. Here it again consists of base paired stem that we can see here. This is the base paired stem and this circular structure which is known as loop. This is known as DHU loop. Here it contains the unusual base. This D represents the unusual base, that is dihydrouridine, and is involved in recognition of tRNA molecule by the ribosomes, just similar to that of T-Cy-C arm. So these both arms helps in recognition by ribosome during protein synthesis. Then the last arm is the extra arm. It is the variable arm. It is not fixed, its size is not fixed. ये vary करता रहता है, and its function is not understood. Next, transcription in prokaryotes. This RNA synthesis always occur in 5' to 3' direction, and this process is completed in 3 steps. First one is initiation, then elongation, and termination. So in case of initiation, this RNA polymerase attaches itself to a specific region of the DNA, which is known as promoter, and forms the initiation complex. And after that, this RNA polymerase moves along the DNA duplex by unwinding it, and one strand acts as a template strand for RNA synthesis up to a termination sequences or termination signal, जहाँ पे special proteins present होती है और वहाँ पे termination होती है, and after that this RNA is released, and this step is known as termination. Next, the enzyme which is involved in transcription process is RNA polymerase. In case of prokaryotes, there is single RNA polymerase which catalyzes all the functions. So RNA polymerase that catalyzes transcription in prokaryotes and is made up of 4 subunits, which is alpha, beta, beta dash, and omega subunits. So there are 4 polypeptide chains which combine to form this enzyme, which is RNA polymerase. The core enzyme binds to a specific sequences on template DNA strand, and that particular portion on DNA is known as promoter region. The binding of core polymerase to promoter is facilitated by a specified factor which is known as sigma factor. So this core combines with the sigma factor to form enzyme which is known as holo enzyme. So prokaryotic RNA polymerase holo enzyme consists of alpha unit, beta, beta dash, and sigma factor. In addition to this, it also contains omega subunit. And what are the various functions of these subunits? Alpha subunit help in chain initiation and interaction with regulatory proteins, while beta subunit helps in chain initiation and elongation, and beta dash subunit helps in DNA binding, and the sigma factor helps in promoter recognition by this holo enzyme. The sigma factor is involved only in initiation of transcription. It plays no role afterward and gets released after initial transcription. So इसका जो main role है, it helps only in recognition of promoter region by this enzyme. After that, it is released from this transcription machinery. Chain elongation is catalyzed by the core enzyme only. So the first step in case of transcription process is initiation. This transcription is initiated by RNA polymerase holo enzyme from a specific point which is known as promoter sequence. Single RNA polymerase found in case of prokaryotes, bacteria, which is called core polymerase, and it consists of alpha, beta, beta dash, and omega subunits. The core enzyme binds to specific sequences on template DNA, and it's called promoter region. The binding of core polymerase to promoter is facilitated and specified by this special factor which is known as sigma factor, and in sigma it is sigma 70 factor in case of E. coli. So this sigma factor helps in recognition of promoter region by this holo enzyme. The core polymerase along with sigma factor is called holo enzyme, that is RNA polymerase holo enzyme. In case of E. coli, promoter consists of 2 conserved sequences, that is 5' to 3' TTGACA at 35 element and 5' to 3' TATAT at 10 element upstream from the promoter region. So this is the transcription site from where the transcription is will begin. And before this transcription site, there is a region which is known as promoter region where the RNA polymerase will bind. Here in promoter region, there are 2 specific sequences upstream from the transcription site, that is 10 base pair upstream, nucleotide upstream from the transcription site. The special sequences are TA, TA, AT, and 35 base pairs nucleotides upstream as TTGACG. So these are some specific sequences which are present in promoter region, and they are recognized by the RNA polymerase holoenzyme. These sequences are upstream to the site from which transcription begins. Binding of holoenzyme to these two conserved sequences of promoter form a closed complex. So this binding of this RNA polymerase to these sequences form a complex structure. Next, the AT-rich region, that is 10 sequences helps 10 upstream sequences helps the localized unwinding of DNA, which is primary requirement for the synthesis of new RNA chain. So the first step is opening of that DNA duplex. It opens from a specific sequences which are AT-rich. It means it contain only adenine and thymine. They contain only double bonds, so it is easy to break these double bonds. And this activity of unwinding is also performed by RNA polymerase itself. Then RNA polymerase starts synthesizing nucleotide. It does not require help of any primase. If the enzyme synthesizes short RNA molecule of less than 10 base pair, it does not further elongates, which is called abortive initiation. So sometimes transcription is halted by the abnormal function of RNA polymerase. If the enzyme synthesizes only short RNA molecule of less than 10 base pairs, it does not further elongate. So that type of transcription is known as abortive initiation. So when the RNA polymerase manages to synthesize RNA more than 10 base pairs long, it ejects the sigma factor. Then after that, the sigma factor is ejected and RNA further elongates and exits from the RNA exit channels. और जो sigma factor है वो core enzyme को छोड़ के बाहर निकल जाता है. Then it is left with only the core enzyme. The holoenzyme gets dissociated into core enzyme and the sigma factor that gets ejected from the core enzyme. This is formation of tertiary complex that leads to formation of tertiary complex. So this RNA polymerase binds to this site, specific site, which is known as promoter region on the DNA. And this sigma factor helps in recognition of these conserved sequences on the promoter region of DNA. Then the second step in case of transcription is elongation. After the release of sigma subunit, elongation of RNA is catalyzed by RNA polymerase in 5' to 3' direction. So this is performed by RNA polymerase. The extension of RNA chain takes place within the transcription bubble. So this leads to formation of this bubble-like structure, which is known as transcription bubble. So this unwinding is done by RNA polymerase itself. The RNA polymerase molecule contains both DNA rewinding activity and unwinds the DNA double helix ahead of the polymerization site and reassociate the complementary DNA strand behind the polymerization site. So both the function, that is opening of this DNA double helix, is done by RNA polymerase itself, and it also helps in this binding of these both the strands again and further unbinding of this DNA double strand in the forward direction in which the transcription process proceeds. So both the functions, that is rewinding and unwinding, is done by this single enzyme, that is RNA polymerase. In prokaryotes, the average length of transcription bubble is 18 nucleotide base pairs. जो average length है transcription bubble की, that is 18 nucleotide base pairs, and about 40 ribonucleotides are added to elongating RNA chain in every second. So हर second में 18 nucleotides add किए 40 nucleotides add किए जाते हैं in elongated RNA chain in every second. The nascent RNA chain is displaced from the DNA template strand. जो नया RNA बनता है वो displaced हो जाता है from this double helical structure of DNA. The synthesized RNA is proofread by hydrolytic editing. So इसमें proofreading भी होती है कि कोई इसमें mismatch तो नहीं बना है. So this proofreading activity is also performed by some enzymes by hydrolytic editing. For this, the polymerase backtrack by one or more nucleotide and cleave the RNA, removing the error and synthesizing the correct one. So this proofreading is also done by this polymerase enzyme itself. So the single enzyme perform all the actions during this process of transcription in case of prokaryotes. Then the third step is termination. It means it is a signal for stoppage of transcription process. So there are two mechanisms of termination. One is the presence of a protein factor which is required for termination, that is known as a Rho factor, and this is called Rho dependent termination. Then the second one is the other types of sequences which results in termination of transcription without any involvement of row factor. So इसमें कोई भी involvement नहीं होती है row factor की. Such sequences are known as row independent terminators. So firstly, the termination is row independent termination in which the row factor is not required. So row independent terminators are marked by the presence of GC rich regions followed by 6 or more AT base pairs with the adenine in 5' end present in the template strand. So जो termination sequences हैं in row independent terminators, they are marked by this GC rich regions. It means there is only guanine and cytosine which are bonded together with the help of 3 bonds. So these bonds require a lot of energy to break it. So these act as a signal for termination of transcription without row factor, which are followed by 6 or more adenine or thymine rich base pairs at 5' end in the template strand. The next, the terminator DNA contains invert repeats which causes complementary pairing as transcript RNA forms a hairpin structure. So ये जो RNA बनता है इसमें कुछ एक complementary sequences present होते हैं which give rise to this structure. Stem और loop की तरह structure जो हमने tRNA पढ़ा था, that forms a structure which is known as hairpin loop. और इसमें जो sequences होते हैं they are complementary sequences. So that results in formation of this structure which is known as hairpin loop structure. The RNA hairpin structure is formed immediately after synthesis of participating region of RNA chain. जैसे ही RNA complete होके बनता है वैसे ही hairpin structure बनता है RNA hairpin structure. The invert repeat is followed by large number of thymine base pairs on template DNA. So जो template DNA इसमें यहाँ पे thymine present होता है. In response to thymine, the uracil appears in RNA. जो uracils हैं वो appear होते हैं RNA के ऊपर. Then the load of hairpin structure is the hairpin structure is very heavy, so it cannot hold the load of this RNA hairpin structure, and it is not tolerated by this AU base pair that are bind together with the help of double bond. So the RNA gets separated from RNA DNA heteroduplex. So इसकी वजह से जो hairpin का weight है वो ये एडेनिन और यूरेसिल के बेस पेयर्स इसको होल्ड नहीं कर पाते हैं और यहाँ से ये नीचे यहाँ से ब्रेक डाउन हो जाते हैं. द बॉन्ड्स गेट्स ब्रोकन डाउन एंड विच लीड्स टू रिलीज ऑफ दिस नेसेंट मैसेंजर RNA in case of prokaryotes. So in this way, row independent termination occurs in case of prokaryotes. Next comes row dependent termination. So in this case, there are some special sequences which are 50 to 90 base pair long and specify RNA transcript that are rich in cytosine residue and largely devoid of guanosine. The row protein binds to elongating RNA chain and moves from 5' to 3' terminus along the mRNA following the RNA polymerase molecule. So जो row factor होता है, these are row factor proteins. ये 5' to 3' direction तक move करती है and they follow the RNA polymerase molecule. When RNA polymerase halt at row-dependent termination sequences, so there are some special sequences which are known as row-dependent termination sequences. Row binds up with this with the polymerase, जो RNA polymerase है ये row factor इसका bind करता है, and pulls the nascent RNA chain from the transcript bubble. और जो RNA है उसको pull करता है बाहर यहाँ से इस transcription bubble से. However, the complete mechanism of row-dependent termination is still not clear. So जो clear mechanism है इसका row factors का which involved in termination of transcription is not well known or clearly known. So this was all about the termination process in case of transcription. So this was all about for our today's discussion about transcription in prokaryotes. If you have any questions, queries, and any suggestions, you can give it in comment section. Thank you. Have a great day.